Thank you to Dr. Ali for inviting me to present at the IBD Journal Club, and thank you for Dr. Moulton for providing expert commentary on our paper that was recently published in Gastroenterology entitled Inclusion of Underrepresented Racial and Ethnic Minorities in Randomized Clinical Trials for Inflammatory Bowel Disease. I'm Nathaniel Cohen. I'm one of the Advanced IBD Fellows at University of Chicago Medicine. Inflammatory bowel diseases have historically been described predominantly in Western countries and in white populations. Having said this, there has been a steady increase in both incidence and prevalence across the globe and in varying racial and ethnic groups, including in the United States. We aim to describe the racial and ethnic representation in IBD clinical trials and whether these changed over time. We systematically reviewed the Phase 2, Phase 3, and open-label long-term extension trials investigating the efficacy and safety of the FDA-approved therapies in IBD. Each publication, supplementary material, table, and abstract was examined for race and ethnicity data. Our search yielded 641 publications, most of which were excluded as they were not randomized control trials or they were Phase 1 studies. Our final analysis included 55 studies of 9 distinct IBD therapies. The range of year of publication was 1960 to 2020. Around 80% were Phase 3 trials and 20% were Phase 2 trials, and the total number of study participants was 24,315. We found that there was a distinct underrepresentation of racial and ethnic minorities in IBD randomized clinical trials, as can be seen from the very low proportions of blacks, Asians, or other patients reported here. Only 60% of the studies included any race demographics, and of these, a third only mentioned the percentage of white participants. Only 10 studies included any ethnicity demographics, and all of these were reported as binary, as in only Hispanic or not Hispanic. None of the studies mentioned limitations surrounding racial or ethnicity inclusion in their discussion section. We did a time trend analysis to assess whether there was any change in terms of racial or ethnicity inclusion in the clinical trials over the last decade compared to prior decades. And while there was a statistically significant increase in both the number of participating continents and countries in this last decade, and also there was a significant increase in race and ethnicity reporting in this last decade, as can be seen from the graph, there was minimal change in terms of the actual racial representation in clinical trials. We can see from this graph that the proportions of whites, blacks, and other patient populations actually remained the same in both decades. There was an increase in the number of Asian participants, proportion of Asian participants, which we felt was probably related to the increased number of clinical trials taking place in Asian countries. The reporting of race and ethnicity and the inclusion of diverse and underrepresented populations in the clinical trials of therapies for IBD and medicine in general actually have been very poor. The participation of Black or African American and Hispanic populations was very low across all the clinical trials, in contrast with the increased incidence of IBD noted in these populations during a similar period. And while Black or African American and Hispanic people comprise approximately 30% of the population in the US, non-Hispanic white people of European descent actually comprise over 90% of clinical trial participants across all fields of medicine. There is great importance in representative inclusion of all racial and ethnicity groups in clinical trials. Not doing so limits the generalizability of clinical trial results to all patient populations. It may affect the ability to ascertain differences regarding drug efficacy, safety, and toxicity, as potential differences in genetic makeup and thus pharmacokinetics are not taken into account. It also highlights the inequality minorities face in accessing high-quality healthcare, particularly in the United States. And it potentially hampers the provider's ability to garner the trust of patients when offering various treatments, and likewise may affect the acceptability of these treatments from a patient perspective. There are multiple possible reasons for the racial disparities noted in clinical trials, and these can be divided into patient-related factors, such as suspicion of clinical trials due to a history of prior exploitation, as has been noted in America as well. Physician-related factors such as certain biases regarding the intellectual ability, education level, the ability to complete clinical trials, or adherence, which some physicians may have. And there may also be structural-related factors such as the economic, educational, and social ramifications of structural racism, as well as the problem of access to quality healthcare, especially centers where these large clinical trials take place. We propose the following action items to try and correct these inequities, starting from the preclinical stages with studies investigating and reporting on racial inequalities to highlight the issue at hand, such as this current study and also the multiple other studies that have been published in recent weeks. Study design, clinical trial design, in order to give adequate power to examine efficacy and safety in underrepresented groups. Looking at recruitment strategies aimed at specifically improving enrollment of groups and strategies to address trial hesitancy. Looking at improving reporting with consistent and standardized reporting of race and ethnicity data across clinical trials, and also a description of the methods used to ascertain race and ethnicity. In terms of the analysis, so to provide subset interpretation of clinical trial data to include minoritized groups. And then post-approval with dedicated and directed communication and follow-up of therapies for underrepresented groups in phase 4 studies. In conclusion, there is significant racial disparity and a very low proportion of racial minorities in the phase 2 and phase 3 clinical trials of therapies for IBD. These disparities are likely due to multiple factors, and we propose that greater effort be made at multiple levels for the appropriate participation of all racial and ethnic groups in clinical trials. We believe that these initiatives will enhance equitability of clinical trials in IBD, build trust between providers and patients of diverse backgrounds, further our understanding of disease processes and response to treatment in different patient populations, and ultimately improve the quality of life for all people living with IBD. I'd like to again thank Dr. Ali for inviting me to discuss our paper, and obviously to Dr. David Rubin, my mentor, without whom this work would not have been possible. Thank you.